Hi, I'm Professor Elaine Holmes, and I run the Centre for Systems and Computational Medicine at Murdoch University in Western Australia. Generally, we've got an interest in a wide range of nutritional areas, and that would go right from being able to assess the quality of food products themselves. So, is olive oil from one particular country or region better than another? What is the chemical composition in different fruits and vegetables, that sort of thing. But moving more from the point of view into the metabolism, what are the metabolites we can detect after eating particular food products, and can we use these as biomarkers of a healthy diet? And I think the approach we take to most problems is a little different from most, because we don't do fruit by fruit or food by food. What we try to do is characterize the impact of a whole diet on metabolism, because people don't eat foods as a single entity. It's always in combination and in a pattern. So we're more interested in dietary patterns and obviously the impact they have on health. So if you think of the cardiovascular disease 10 years ago was very low, relatively speaking, in China and Japan, in the Asian countries, but very high in America. So one of the questions was, it can't be the genetics, because the genetics has been the same way for decades and hundreds of years. So it has to be something to do with lifestyle, and the most likely culprit for poor diet and bad consequences on cardiovascular health is going to be the different diets, the unprocessed, the sort of processed foods, the carbohydrates that are heavy in the American diet. So we started looking at diets and their relationship to blood pressure in different populations, and we moved from there to saying, well, okay, what does a healthy diet look like, and is a healthy diet in one person the same as a healthy diet in another? If everybody eats a healthy diet, do they all respond the same? There's a lot in the literature that was starting to come out to suggest there's not one size doesn't fit all. Some people respond differently to different diets. So we started to go down that route, and that's very much been our focus in the last year or so, trying to predict which sort of diet is best on an individual basis. So NMR's really useful tool in that it's highly reproducible, and it may not be as sensitive as mass spectrometry, but the chemicals you see in a urine or plasma profile pretty much reflect the human endogenous metabolism. So you can see things like energy metabolism. They also can reflect the dietary compounds that are either unchanged and excreted or metabolized. So for example, you can pick up proline betaine, which is a mark of citrus fruits, or there's some sulfur containing metabolites that would come from cruciferous vegetables, carnitines from meats, and so on. So you can actually detect chemicals from the food itself. And then the third piece of information that NMR is very good at giving you is the impact on the gut microbial metabolites or host microbiome metabolites. So things like hippurate, paracresol sulfate, phenylacyl glutamine, these are all metabolites that are made that are made by the gut bacteria and may be modified after by the human body, but they all have a gut microbial component in the metabolic component in them. So we can gauge the impact of the diet on the microbiome and gut health as well, which has a more global impact on full body health. So NMR is very attractive because you can get these three pieces of information, and it's a snapshot. You get it in 5 minutes, it's rapid, and that lends itself to doing metabolic profiling, snapshots over weeks, months, years, so that an individual can follow their metabolism and see if they're doing well on a diet or not doing so well, and they can see how they're doing. And yeah, it's efficient, it's cheap and robust. Basically, there's no one body fluid that would give you the whole answer, but if I had to pick one, it would be urine, because urine isn't held under such tight homeostatic control as plasma, for example. So it carries waste products, and these are present in orders of magnitude more than perhaps you would see them in the plasma. So you can detect a whole range of chemicals that you don't see in the plasma. And also, if you're thinking about gut microbial metabolites, you might think that fecal water is a better biofluid, but actually, because a lot of the chemicals are reabsorbed, they reappear, and they're low molecular weight, they are more stable in the urine. So the gut microbial metabolites coming from intestinal metabolism, much more, the bacteria in the intestine are much more stable in the urine than they are in the feces, which is prone to a lot of degradation. So even for the gut microbiome metabolites, I would go for urine. Of course, if you're looking at things related to cardiovascular and cholesterol metabolism and things like the lipid lipoprotein profile, would also give you a very good bang for your buck in terms of trying to profile. But if I had to pick one, it would be urine. To me, the main advantage of NMR is the ease of use and its broad applicability, and also its robustness. There are specific things that you can, or pieces of information you can draw from NMR that you couldn't, for example, draw from mass spec. It's much more atom-centered information. And one example I can think of off the top of my head is the N-acetyl glycoproteins, or GLIC-A, which is a marker of inflammation. There are pulse sequences you can apply, diffusion weighted pulse sequences you can apply to the spectrum to just focus on the different types of glycoproteins, so GLIC-A, GLIC-B, and these tend to correlate with inflammation. So you can get emotional and relational information about molecules from NMR that you can't draw from mass spec. So on a day-to-day basis, though, it's simply the efficiency of NMR that makes it such a good tool. Again, we have lots of goals and lots of different projects, but the most exciting one, I think, is to be able to really drive personalized nutrition. So to be able to pair the technology with dietitians, use it in clinical practice, so that just like your GP or dietitian will ask for bloods to be run for glucose levels or CRP, I'd like to see us get to a point where they would ask us also for a profile to see what the particular dietary recommendations are doing for their patient, and use it as a way to track that patient over time. So really trying to integrate the NMR metabolic profiling into clinical practice associated with the whole dietitian process. So it would be really useful for people who wanted to lose weight, for example, but also not just for that, but also for people who are pre-diabetic, heading towards diabetes, or who have cardiovascular disease, maybe even during pregnancy. There are lots of windows where dietary profiling would be really useful in a clinical scenario. Well, our approach has very much been to collaborate with the big population studies. So we had one collaboration with the Intermap, which was 5,000 people sampled twice, and that was across the US, the UK, China and Japan. And then we have other studies like OmniHeart, which is a US-based study, Intersalt, which is a 17-country study around blood pressure. The Busselton study, which is our Western Australian study on healthy aging. The idea is to get as many population studies as we can, and to really learn about the impact of the diet. Of course, that also involves these studies having good dietary records. And we already know that sometimes dietary records aren't the most reliable, because human beings have a tendency to underreport or overreport, depending on whether you're looking at bad or good foods. But where we have good data, we would like to we take these population studies and we try and match them to different regions, different ethnicities, so we get a good global profile of different diets. Okay, so I think one of the most interesting things we found is that we suspected that one diet doesn't affect everybody in the same way. But when we looked more closely into why that was, and going across multiple studies, multiple population and clinical studies, what we started to see was that the difference between people was largely due to gut microbial metabolites. So, for example, people in one of the studies that was run by Isabel Garcia Perez at Imperial was where she put 19 people into a metabolic unit. So these people were followed all day, every day, 24 hours. Everything they ate, every action they took was on camera, on video. So we knew exactly how much exercise these people had had, what they'd eaten, and they had exactly the same. And yet some people excreted 5 or 6 calories, habitually excreted fewer calories in their urine than others. And also, if you look at the NMR profiles, different pathways were lit up. So some people just seem to have a slightly different metabolism. And a lot of that difference in metabolism was coming from the gut bacterial metabolites. So then you can start to ask the question, well, is that dynamic or not? Does one person have one type of gut microbiome for the whole of their life? And is this what you are with respect to how you metabolize a diet? Or what if we could actually use some sort of prebiotic or probiotic and try and change the gut bacteria? Can we give you a gut bacteria that's less efficient at harvesting calories, for example, if people are overweight, or more efficient if they're underweight? And can we start to play around with that and modify diet? So I think that was the most interesting thing we found so far. But there's a lot of scope for this approach in all sorts of areas, and even down to our laboratory studies where we're profiling postdocs, you'll find that some people will have one food that they metabolize totally differently. Like we have somebody in our lab who, if they eat bananas, we can't see any banana metabolites, but they lose all their Krebs cycle intermediates in their urine, which is really strange. And it happens, we've repeated the experiment. So some people have just got an odd metabolism with respect to processing diet. Regardless of how you metabolize something compared to another person, if you put somebody on a good diet with lots of fruits and vegetables, following WHO healthy eating guidelines, then you're going to be better off than following a fast food diet. So if you looked at the lipoproteins, you'd see your cholesterol lowering, your HDL going up, your LDL going down. You'd see some good response in any case. So the differences between people I'm mentioning are just that, they're differences between people, but in terms of how do you respond to a diet, then you would expect a healthy diet to have a good effect in most people. It's not so different that some people shouldn't eat a healthy diet. We have lots of ideas. Definitely we want to get more into the personalized nutrition, and for that reason we formed the company Melico, which is short for Metabolic Life Coach, and it's trying to implement an NMR profiling strategy into the pipeline for clinical dietary advice. But we want to go a step further than that, and in Australia we've had the opportunity to work with somebody in the agricultural industry, and the idea here is to put him and a chef together, and our technology, our models together, based on the NMR profiling, and say, well, okay, what's your starting profile? What's your baseline? How do you look like? And then by trying out various diets and various people, so match a person to a particular meal. And then for those people who don't like cooking and can't be bothered to work things out for themselves, have various sort of ready meals that are, because they're made by a chef, aren't just bland, but have, that actually tasty. So not only are they good for you, but they taste nice. And to be able to implement that as a whole package, going from farm to health check, if you like. So that's going to take a little while to work up, and we've got lots of clinical studies planned along those lines, but that's kind of a fun and exciting project at the minute.